It's Ingrid and my pleasure to welcome you all to the 2026 webinar series for the Transparent Cost program. So we're very excited that Catherine Hogan is going to launch or kick off this year's webinar series. As you know, we'll have 10 webinars over the course of the year. We won't have one in August or in December. So we will have 10 programmed for this year, which will focus on all the different work packages, primarily work package 2, working group 2, 3, and 4. So today we have Catherine Hogan with us. She's a lecturer at the European Cancer Stem Cell Research Institute, School of Biomedicine, Cardiff University. She's been there since 2020. She's had a fantastic career endeavors. She did her PhD at the Randall Division of Cell and Molecular Biophysics in King's College London, and completed her postdoctoral studies with Dr. Fujita in the Laboratory of Molecular Cell Biology at UCL. And then in 2013, she joined the European Cancer Stem Cell Research Institute as a research fellow, where she established the pancreatic cancer research program that she leads, where she combines in vivo models of pancreatic cancer with organoids, quantitative 2D, 3D imaging, and transcriptomics, among many other things. So the goal of the Hogan Lab, according to Catherine, is to unravel the mechanisms underlying how pancreas epithelial tissues stay healthy following stochastic low-level genetic insults, and determine how these processes are deregulated during early tumorigenesis, which I think is a fantastic point of view. We need to really understand what's happening early on, and Catherine will give us a glimpse into the many things she does in her laboratory. So again, Catherine, both Ingrid and I thank you for joining us today. We have almost 40 people, a lot more people will join, so the stage is yours.
Thank you very much, Bruno, Ingrid, and to the Transpan community for this invitation. I'm really delighted to have the opportunity. I'm going to share my screen, and hopefully you can see my screen now. Yeah, that's okay.
Yes.
Okay, great.
Yes.
Fantastic. So we'll get started. So yeah, thanks for the kind introduction, Bruno. I'm going to give you an overview of some of the research we've been doing at Cardiff since I started my lab, really, looking at how adult pancreas tissues stay healthy and how they counteract really spontaneous genetic mutation that can cause cancer. And we believe that our work really has huge implications for understanding how pancreatic cancer really starts. And that's one of our endeavors. So we know that pancreatic cancer starts with KRAS mutations. So pancreatic cancer is primarily an adult tissue, and it arises in the epithelium of the adult pancreas. Let's see if I can get a pointer. Predominantly acinar cells, but we also know that ductal epithelial cells can also become transformed to initiate cancer. And we know that oncogenic KRAS mutations are the key driver event, and also work from many others in over the years has shown that KRAS mutations are also essential to drive tumorigenesis. Hence the clinical endeavors now to drug KRAS signaling. And we're really interested in this very early stage. So how does this normal epithelium become transformed and metaplastic to then drive these precursor lesions, which can come in two main flavors? One is the pancreatic intraepithelial neoplasia, or PANINs, but we know that KRAS mutations, depending on the cell of origin and the combination of other mutations, can also drive cystic pre-malignant lesions. We also know that pancreatic cancer is very heterogeneous and it's very plastic, and not all PANIN lesions or all cystic lesions will develop into tumours. And we're also looking at why this might be the case, but I'm not really going to talk about that today. But one thing I would like to say, just as an aside, I suppose, or as a comment, is that pancreatic cancer is very heterogeneous in terms of the tumour as well. So we know that it has classical epithelial phenotypes, but it can also take on hybrid fates, which are this classical mixed with this EMT or plastic-like state, basal-like state. And how this heterogeneity is really controlled is also very poorly understood, but is a big endeavour of the field. So as I said, in my lab, we are really interested in very early stages. And we've started with the premise that KRAS mutations arise spontaneously in the tissue. So they arise in epithelial cells that are present then in the tissue in very low numbers. And we model this in my lab using a variety of approaches, which I'll describe today. And of course, this spontaneous mutagenesis then comes with the assumption that these KRAS mutant cells have to be able to grow and expand within the confines of a tissue that is otherwise under very tight homeostatic controls, because it is otherwise healthy. So we're asking two questions of the system. The first is, how does the adult pancreas maintain health? So how does it stay healthy over the course of the adulthood, despite the fact that spontaneous KRAS mutations can arise? And we know that this is important because pancreatic cancer still remains to be relatively rare within the human population. And the flip side of this, of course, is how do KRAS mutant cells expand and grow, as I say, in this confines of a normal healthy tissue. So we believe that by understanding the fundamental biology underpinning these two questions, we will gain new knowledge and information on how pancreatic cancer really starts. And we hope that this can feed into new early detection strategies, such as the search for new biomarkers, but also potentially into new prevention strategies. So this work really started when I was a postdoc, and I should say a disclaimer that I'm a cell biologist by training, and my lab is really looking at fundamental cell biology. And when I was a postdoc in Yasu Fujita's lab, we asked the question of what happens if RAS mutant cells are present in tissues in low numbers. And to do this, we engineered MDCK epithelial cells to express GFP-tagged oncogenic RASV12. And this is under the control of a tetracycline-inducible promoter. So it allows us to create monolayers of epithelial cells where RAS cells are present in the tissue in very low numbers. So we can seed RAS cells in with normal healthy cells, wait for proper cell-cell adhesion to occur, and then add our tetracycline to induce oncogenic RAS expression. So this is a movie of one of those experiments, and hopefully it'll go back to the start soon, but you'll notice at the start that maybe I can come back and go out again, that this cell here and this cell here, which we have pre-labeled with a fluorescent dye, is going to express oncogenic RAS. And over the time of the course of the movie, you'll notice that these RAS cells, they initially proliferate, but then they become very small in shape. So they shrink, they lose volume, they become contractile, and they are eventually extruded out of the epithelium. So they're kicked out of the epithelium by these normal neighboring cells. And this is occurring from the apical side. This is apoptosis independent, as you can see quite clearly that the RAS cells then start to proliferate and form this kind of aggregate of cells that loosely kind of attaches to the underlying normal cells. So when I started my lab, I was pursuing what really triggers this elimination mechanism. And we decided to come at it with the idea that something is expressed at the cell-cell contact between normal cells and mutant cells that then triggers these events. And so we decided to come at it with a different approach and essentially collide sheets of MDCK cells together. So here we've got GFP expressing RASV12 cells colliding with non-labeled normal MDCK cells. And this is essentially a wound healing assay. And what we found was that RAS cells take on this very distinctive morphology again. So upon contact with normal cells, they become very round and contractile, and they segregate away from the normal cells by a very clear and distinct boundary. And of course, as our controls, if we collide sheets of RAS cells with RAS cells, or if we incubate pre-labeled RAS cells with RAS cells, we don't see any of these phenotypes. So this tells us that direct interaction with normal cells is really causing this effect. We were also able to show that this is in flies, so using Drosophila melanogaster, but also in pancreas ductal epithelial cells in primary culture. We see that if we co-culture RAS-expressing cells with normal cells, these cells can be extruded, and they also tightly pack together to form these very contractile tiny packs of clusters of cells. So this is reminiscent of a process called cell competition, which is essentially a quality control system that's very well documented in the Drosophila melanogaster field, but has been in the last 10 years really shown to play a very important role also in mammalian tissue health. And just couple of key take-home points about this is that so cell competition is really context dependent. It really depends on the genotype that the cell is expressing relative to the genotype of the neighboring cells. So, for example, a genetic mutation that can be cancer-causing will autonomously give cells an advantage in that they become hyperplastic, but this genetic mutation could render the cells to become losers in a competition scenario if they're in the context of normal wild-type cells. So, for example, in this here, these scribble knockdown cells are then triggered to die by apoptosis and are eliminated, or they can be eliminated by cell extrusion. If this is happening in a stem cell, then the stem cells are triggered to differentiate and exit the niche. And so it really is about the fact that cells are able to monitor this relative fitness, which is a very broad term, to each other, to their neighbours. And cell competition became quite interesting to the cancer field about 15 years ago or so, because it was realized that some cancer-causing mutations, such as oncogenic MYC or loss of function of APC, can render cells to become what are called super competitors. And that means that they can stay in the tissue and they can expand in the tissue by killing off the normal wild cells. So either they trigger the normal cells to die by apoptosis, or again, if this is occurring in a stem cell niche, then they retain that stem and colonize the niche by triggering the normal cells to differentiate. So again, when I started at Cardiff, I was very interested in understanding how does this relate to a kind of a human relevant model of disease when you express oncogenic RAS mutations in cells in low numbers. And pancreas was an ideal opportunity for this because of the fact that oncogenic RAS mutations are those key driver mutations in pancreatic cancer. So when I started the lab, we decided to do this and dive into the mouse models, and we use the KC mouse model, and this was in collaboration with Jen Morton at CRUK Scotland. And as you know, the KC mouse model drives oncogenic KRAS G12D in cells that express PDX1 promoter. And we use the CRI-ERT model because this allows us to express the oncogene in adult tissues, but it also allows us to have a level of control in the expression by tamoxifen. And at the same time, our cells are expressing an RFP reporter that's coming from the ROSA locus, which allows us to trace the cells. So we designed a low-dose tamoxifen model, which is what we continue to use from here on in. And this is where we titrated the dose of tamoxifen to a very low level, which allowed us to generate tissues that are basically mosaic for oncogenic KRAS expression and RFP. So we have recapitulated what we've done in the MDCK cells, essentially. And of course, in this scenario, the interaction with normal cells is quite high. And our control setting was using what we call a megadose tamoxifen, which is essentially the dosing regime that people will use in the field to switch on oncogenic RAS in adult tissues, where most of the cells are expressing the oncogene and the RFP reporter. And of course, in this scenario, then interactions with normal cells will be very low. And this is a typical representative confocal image of what we see in adult tissues following KRAS are following low-dose tamoxifen. So you can see that we have RFP expression in acinar cells predominantly, but we also see a very large level of recombination in islets, not surprisingly, and we also see it in the ducts. So I'm going to just move my screen, this out of screen so I can see my pointer. Okay, so that tells us that we've got really nice kind of mosaic salt and pepper kind of tissues, and there is a level of stochasticity in there as well. So what happens when we monitor these cells over time? And to do this, we basically take a tissue section from our mice that have been harvested at 2 time points, 7 days post-tamoxifen induction or 35 days post-tamoxifen induction. I should say that we are inducing adult mice, so 6 to 8 week old mice, predominantly 7 to 8 weeks. And then we take a tissue section and we image it under a confocal microscope, and we then quantify the amount of RFP that's expressed in the tissue and express it as a percentage of tissue area. So at low dose tamoxifen, we're getting recombination in around 20% of the tissue, and this remains quite stable over time in wild-type control tissues. So there's no significant change in the level of RFP over time. And again, this isn't too surprising, but quite reassuring, knowing that pancreas tissues are very slow proliferating tissues, and so the turnover of the RFP positive cells is very slow. But if we look at our tissues that are now expressing oncogenic KRAS mutant cells, we can see that we see a significant loss in the number of RFP in tissues over time. So from 7 to 35 days, there is a dramatic decrease in the percentage of RFP. And that it indicated to us that KRAS mutant cells could be lost from the tissue. Of course, a very important control experiment was to do the megadose, as we call megadose experiment. So here we've used the high dose tamoxifen to recombine most of the tissue. Now we're seeing an RFP expression at around 80% in the tissue. And now we see no loss of our RFP over time in cells in tissues that are expressing oncogenic KRAS. So this indicated to us that this loss in the RFP is really happening because of the fact that RAS mutant cells are present in low numbers and are directly in contact with the normal cells. So this is predominantly representative of the acinar compartment, we would argue, and so we went on to show that ductal epithelial compartments are very quickly and very efficiently getting rid of mutant cells, and this is happening very early as 7 days. And islets are also able to eliminate oncogenic mutant cells from the compartment. So what's triggering this RAS cell elimination from the epithelium? And this was something we were very interested in. And based on the fact that in each of the epithelial cell systems that we looked at, RAS mutant cells always adopt a very conserved morphology when they're surrounded by normal cells. So they become very tightly packed together. We can show this by counting how close the nuclei are together, let's say in a cluster. And they also segregate, as I said, away from the normal cells. And this was very reminiscent to us of cell repulsion, which can be controlled by a family of repulsion factors called EPH-ephrin family. And we decided to go after this because we knew that EPHA2, which is one of the main receptor tyrosine kinases expressed in epithelial tissues, is a MAP kinase target for RAS. So it's a RAS target, transcriptional target, and indeed we can show that in MDCK systems, our RAS mutant cells upregulate the expression of EPHA2, and when they're surrounded by normal cells, they activate this receptor, as we can see by phosphorylation. So we knock down the EPHA2 receptor in the RAS cells, and we no longer can see this segregation or elimination phenotype. And in fact, the RAS mutant cells take on this kind of very mesenchymal-like morphology. And of course, we lose activation of FA2 receptor. So we were able to show that this is evolutionarily conserved in flies. So if we express small clones of GFP RASV12 cells in the wing imaginal disc, then they will segregate away from normal cells and be eliminated from the apical side of the tissue. And knocking down EPH receptor, flies only have one, using either RNAi or a dominant negative, we lost this segregation and elimination phenotype, telling us that EPH receptors are also required in vivo. But of course, the crucial experiment was to do it in the mouse. And for this, we crossed the KC mouse with an EPHA2 loss of function. And this is it's not a genetic null, but it's a loss of protein function, which is constitutive in the tissue, which we termed KCE mice. And indeed, what we see is that there is no loss in the RFP coming from these tissues, suggesting to us that this EPHA2 receptor is essential and is conserved in evolution to eliminate these KRAS mutant cells. So how is this really happening? And this is something that we're still working on. We don't really fully understand it. But to get a little bit of insight into the cell biology, I just wanted to point out a couple of things which will be important for the next part of my talk. So in my lab, we use a lot of 3D, quantitative 3D imaging. Here there's a video of basically a ribbon, so we can basically fix our tissues and embed them in plastic, which allows us to do cut ribbons as you would for electron microscopy, for example, and then build these 3D rendered stacks of the tissue. And that allows us then to extract cell descriptors and information about the cells using Amaris, for example. So what we noticed was that when RAS mutant cells were surrounded by normal cells, they became very round, as I said before. They cluster very close together, and they also lose volume. And this is significantly different compared to controls. But this volume change was no longer seen when the cells were present in EphA2 knockout tissues. And what's even more interesting is that if we looked at the volume of the cells that are immediately adjacent to the RAS mutant cells, then these cells are gaining volume. And we think that this is because they basically don't want to, are trying to mitigate any loss of barrier function. So while RAS cells are losing volume, the immediate adjacent neighbors are gaining volume. And again, this is mitigated or completely obliterated when you knock out EphA2. So we think EphA2 is really required for cells to be able to remodel their shape and their volume. And how they're doing this, is still under investigation. Another thing that we noticed in this model is that E-cadherin is also being modulated in an EphA2 knockout fashion. So this is using either pancreatic ductal epithelial cells that are coming from the mouse, or in vivo fixed tissues, either looking at the exocrine compartment, and we've also done it for the islets. And what we noticed is that when RAS mutant cells are, again, present in the tissue in low numbers, and if we look at the junction that's between the RAS mutant cells, which are labeled here in RFP, and the non-labeled wild-type neighbors, we found kind of a robust punctate staining for E-cadherin. And this suggested to us that E-cadherin is being turned over. It's not very stable, potentially, at this junction. In fact, if we look at this in primary culture, we could see there was a significant increase in the amount of E-cadherin that's endocytosed in the cells. So again, suggesting that you need to be able to turn over your E-cadherin at the boundary between RAS cells and normal cells to be able to be segregated and eliminated. And again, if we did this in an EphA2 knockout context, so either in vivo or in vitro, we could see that the E-cadherin now became much more stable. And we could score this by scoring the amount of E-cadherin fluorescence at the cell-cell contacts. So we think that EphA2 is required to trigger this turnover of E-cadherin. And if it's not functioning, then E-cadherin remains stable at the junction. And this stability is probably feeding into the way cells can also remodel shape and volume. But one kind of take-home message from this, I guess, is that to be eliminated, you need to be able to remodel your junctions. If you're unable to remodel your junctions, then you're probably not going to be eliminated. And this is a consistent message for the next half of the talk. So what does it really mean for cancer, and what does it mean for the tissue health? And what we were able to show in this study is that clearance of oncogenic mutant cells we deem to be tissue protective. And that's because if we score the number of pannin lesions that are present in the tissues at this 35-day time point, they're actually very rare. We rarely see them. But if we do the same analysis in this EphA2 knockout, these KCE mice, we increase the frequency of these pannins. And of course, we're scoring pannins here using Alcium blue as a readout. And this suggested to us that eliminating mutant cells basically reduces those kind of oncogenic niches or those oncogenic hotspots that allow the tissue, would allow mutant cells to transform into a pannin. And by knocking out EphA2, you're basically reversing this. You're increasing the number of niches that are in the tissue that can go on to form pannins. So EphA2 dependent clearance of these KRAS mutant cells reduces pannin lesion formation and therefore is tumor preventative, we would argue. But of course, one other very important observation from this study was that the cell elimination mechanisms are inefficient, and you may have observed this all the way through. So the pancreas can get rid of the mutant cells, and it does get rid of maybe half of them, but there is a very considerable population of cells that are left behind in the tissue. So what is it about these cells that allows them to stay in the tissue and be protected from this elimination mechanism? So just to summarize this part, I hope the data has convinced you that KRAS mutant cells can be outcompeted and cleared from adult pancreas in vivo. And in fact, in every epithelial cell system that we've looked at, if RAS mutant cells are present in tissues in low numbers, they can be eliminated. Adult tissues can get rid of the tissue of the cells from exocrine and endocrine compartments, and EphA2 receptor tyrosine kinase is essential, even though we don't fully understand the mechanism underlying it. What we do know, though, is that it definitely regulates E-cadherin-based cell adhesion, and we think that's important for the cells to be able to dynamically change their shape and volume, not just in the mutant cells, but also in the neighbouring adjacent cells to be eliminated. And finally, this process is tumour suppressive, we would argue. So it helps to promote tissue health by getting rid of these mutant cells, protecting against cancer initiation. And of course, the take home from this is that overriding this process must be essential, therefore, for cancer initiation. So how is it that these cells that are left behind get to stay behind? And that's the work that we've been doing in the last 5 years, which was recently published last year. So I'll just give you an overview of the work. And this was work that was done by a postdoc in my lab, who's now a junior PI or junior fellow, Pancreatic Cancer Career Foundation fellow in my lab, called Bea Salvador. And Bea decided to take this approach by using, we decided to use 2 mouse models in this case. So we're now using the KC mouse model, but we also decided to look at the KPC mouse model. And the rationale for this was that potentially if you have a second mutation, then you're going to become a super competitor. And that was the original hypothesis. So as just to remind you, this again is using the PDX1-CRE ERT to activate expression of oncogenic KRAS G12D in cells, or this P53R172H mutation, which would be then KRAS wild type, which we term PC, or the cells express oncogenic KRAS plus this P53R172H mutation, which is the KPC model. So again, we do the same experiments inducing adult mice with low-dose tamoxifen and then harvesting the tissue over a fixed time point, 7, 35 and 70 days post-tamoxifen induction. So the first thing Bea did was just to confirm that cells were being eliminated from the tissue. And I suppose reassuringly, she was able to confirm this phenotype. So KRAS mutant cells are eliminated from the tissue over time. P53 mutant cells, which are KRAS wild type, are also eliminated from the tissue. But if the cells express both mutations, they're not eliminated at all. And we see no loss in the RFP over any time point. So having that second mutation definitely gives those cells an advantage, a survival advantage at least. And Bea was able to show again that this is happening in all of the different compartments. So ductal epithelial cells can clear single mutant cells, but not double mutant cells, and similarly with the islets. So our hypothesis then was maybe there is a common signature or there's a common signaling pathway that's controlling how all of these cells that are not eliminated function, or how they get to survive in the tissue. So to test this, Bea decided to FACS sort RFP positive cells, and she enriched for acinar cells in the protocol using a leptin antibody, and then pooled these cells and we put them through bulk RNA sequencing. So we're comparing KRAS mutant alone P53 wild type to controls, P53 mutant KRAS wild type to control, and then the double mutant cells to controls. And this is a gene set enrichment enrichment scores for our bulk RNA sequencing results. And just again, to give you an overview of the data, so KRAS mutant cells predominantly upregulate genes, whereas P53 mutant cells predominantly downregulate genes. And this is also true for the double mutant cells. So we think that P53 mutant cells are really switching off a lot of pathways. Which was something which was quite surprising to us, is that we didn't see one main, common gene sets that were standing out, but we did see some common features. So, for example, KRAS mutant cells will upregulate P53 pathway, and P53 mutant cells seem to downregulate KRAS signaling. So having aberrant KRAS signaling and P53 signaling seems to be very important for staying in the tissue. We also noticed a couple of things in common in that KRAS mutant cells seem to be modulating Wnt signaling, particularly Wnt5a ROR signaling pathway, and p53 mutant cells were downregulating canonical Wnt beta-catenin signaling and also downregulating MYC targets. So we decided to focus on Wnt pathway. So considering that, so first of all, we decided to look at whether these cells that are left behind in the tissue have tumorigenic signatures or PDAC signatures. And again, what was quite surprising was, but maybe reassuring also, is that KRAS mutant cells that are retained in the tissue will strongly correlate with pancreatic ductal adenocarcinoma signatures. So even at this very early time point, while they're present in the tissues, they already have tumorigenic potential. But surprisingly, the p53 mutant cells, particularly the double mutant cells, are not switching on pancreatic ductal adenocarcinoma signatures at this time point. And of course, if we age these mice, they'll get PANINs in the KRAS context, not in the p53 context, as you'd expect, and our KPC mice will eventually succumb to PDAC. And this is the work, just to summarize, Bea has now been taking this forward a little bit to look at whether these cells have tumorigenic potential if you add chronic pancreatitis. And she's shown that these cells, KRAS mutant cells that are left behind in the tissue, once they're hit with chronic inflammation, can progress through PANINs to malignant disease. So these KRAS mutant cells that are left behind in the tissue can initiate cancer. So let's have a little bit look at more at what the kind of defines these cells. And we looked at the gene signatures and focused predominantly on KRAS mutant cells that are left behind, and because this is much more physiologically relevant as well. And what we found was that KRAS mutant cells will have stem-like signatures, so they strongly, significantly upregulate stem signatures. So, for example, the embryonic stem cells, ramalostemness, etc. And they also upregulate pancreas progenitor gene signatures. They also upregulate metaplasia, as you'd expect. So we saw strong signatures for SPEM, which is this gastric pylori phenotype. We also saw evidence for ADM. And at the same time, we noticed that the cells are upregulating cell dormancy signatures and diapause signatures. And I'm just showing one of those signatures here. So this was quite interesting, but also not surprising, because we really don't see any proliferation in the tissue at these time points. So in fact, if we go all the way out to 100 days, there's very little proliferation in the tissue unless they have become a PANIN. KPC cells, similarly, they don't really proliferate until they get to become malignant. So this dormancy feature was quite nice to see, and Bea used P27 as a marker of cell cycle arrest and showed that KRAS mutant cells have much higher expression of P27, or the number of P27 positive cells are higher in tissues expressing retained oncogenic KRAS cells. So I mentioned that the cells are upregulating, we think, Wnt5a ROR pathway. So we decided to focus on this a little bit more. And just to confirm that the Wnt pathway was upregulated in retained KRAS mutant cells at the gene setting. And we noticed that beta-catenin dependent Wnt seemed to be downregulated. So we didn't see a signature for beta-catenin signaling. The kind of classical targets were turned off, like MYC, axin 2, etc. Whereas when we look at Wnt5a ROR pathway, which would be the non-canonical Wnt pathway, we do see an upregulation of Wnt, non-canonical Wnt signatures in KRAS cells that are retained. And indeed, these cells will have a higher level of Wnt5a at the messenger RNA level. And also, Bea was able to score a higher level of Wnt5a fluorescence in tissues that contained oncogenic KRAS mutant cells compared to controls. So let's go back to the cell biology, and we decided to explore this a little bit and ask, well, how is Wnt signaling working? So firstly, we confirmed that we could see some evidence of Wnt signaling in the tissue. So using CD44 as a readout for Wnt, we could see a strong upregulation of Wnt, CD44 at cell contacts in the tissues, and Bea was able to score this. If we look at beta-catenin, we didn't see any beta-catenin in the nucleus of any of the cells, again, kind of substantiating this idea that non-canonical Wnt is probably not active. But what we did notice was that there was a slight but significant increase in beta-catenin at the cell-cell junctions between the RAS mutant cells that are RFP labeled and the non-transformed cells. And if we look at E-cadherin, we also see this increase in E-cadherin expression at the junction between the KRAS mutant cells and the normal cells. So this was very convincing for us because it was again reminiscent of what we'd seen in the EphA2 knockout, where cells are not eliminated if they can stabilize their junction. So we decided to look at this a little bit further. So to look at the functional relevance of this, Bea went back to the MDCK epithelial cell system. So here we're co-culturing GFP-tagged oncogenic RASB12 expressing cells with normal MDCK cells in a monolayer. And these monolayers are seeded on collagen 1, so they are nicely polarized. And in this case, what Bea looked at again was beta-catenin and E-cadherin at the cell-cell contacts. And I think what you can see quite nicely is that when the cells were treated with Wnt5a, you get this real stability of the cadherin and beta-catenin at the junctions. And this is not seen when cells were treated with Wnt3a, which is deemed to be a beta-catenin dependent Wnt. And she was also able to score this looking at the level of fluorescence at the junction. And if we looked at this a little bit more closely, we could see that basically when RAS cells are surrounded by normal cells, you do get punctate E-cadherin. And we'd previously shown that if you knock out E-cadherin, you don't get any elimination either. So we know E-cadherin was required, and you do get this turnover of E-cadherin in epithelial cells, RAS epithelial cells that are deemed to be eliminated. But when these cells were treated with Wnt5a, we saw a significant downregulation in the level of internalized E-cadherin, and we could reverse this using Wnt inhibitors. So this is using a porcupine inhibitor to block Wnt signaling, and we could reverse the number of punctae that had been, we could increase the number of punctae that were internalized for E-cadherin when treated with the Wnt inhibitor compared to Wnt5a alone. So we think that Wnt5a is reducing E-cadherin turnover, potentially through endocytosis, but we just haven't confirmed that yet, which allows for RAS mutant cells to be eliminated from tissues. And so if you block this endocytosis and turnover, you're much more likely to be retained. We also showed this through pancreatic ductal epithelial cell culture. So this was one of the reviewers' comments, and we went back to primary culture where we could co-culture KRAS expressing cells with non-transformed ductal epithelial cells and put them in the presence of Wnt5a or in Wnt5a plus porcupine inhibitor. Somebody has their microphone on. Oh, there we go. And so what we found was that Wnt5a will basically increase the number of cells that are retained within the monolayer. So here we're looking at integrated cells, so the cells that are still present in the monolayer at the same with the non-transformed cells. And if we block Wnt signaling, we could reverse this. And we also were able to show that endocytosis of cadherin is also happening in this cell system, where Wnt5a is decreasing the number of internalized E-cadherin, and this could be reverted with a porcupine inhibitor. So Wnt5a is promoting stable cell-cell adhesions in primary pancreatic ductal epithelial cells as well. So what about function? And to test function, Bea again started with MDCK epithelial cells and started to score the number of cells that were extruded or non-extruded from the monolayer. So here we're presenting non-extruded cells. So in other words, cells that are retained in the monolayer, and you can see that treatment with Wnt5a significantly increases the proportion of cells compared to PBS, but also compared to Wnt3a, which we know to be a beta-catenin dependent Wnt. So treatment with Wnt5a keeps cells in the monolayer, and we could reverse this using either porcupine inhibitor or a frizzled antagonist. And in the presence of either of these inhibitors, then the number of cells that were not extruded reverted to below controls. And that means that actually we were now triggering the elimination of RAS mutant cells from the monolayers. But of course, the key experiment really was to understand whether this had any requirement in vivo. And so what Bea did was take the KC mouse or the PC mouse, treat with low-dose tamoxifen, wait for 5 weeks for the cells to be cleared from the tissue, and then treated with porcupine inhibitor for a period of 28 days, and then looked at the level of RFP that was remaining in the tissue compared to vehicle. And she showed that you see this significant decrease in the number of RFP positive cells in KRAS expressing tissues, but also in P53 expressing tissues when treated with porcupine inhibitor. So this suggests to us that treatment with porcupine inhibitor inhibits this Wnt signaling that is keeping the cells in the tissue, allowing for these cells now to be eliminated. But obviously there are cells that are Wnt independent, and we are currently exploring what those could be. We also Bea also tried to look to see if there was a connection between Wnt signaling and dormancy and Wnt signaling and stemness, and she found that in Wnt inhibited tissues, there was again a small but significant decrease in the number of P27 positive cells, suggesting that Wnt may act upstream of cell dormancy in KRAS mutant cells. And again, using SOX9 as a marker for stem or reprogrammed cells, we saw again a small but significant decrease in the number of these cells in Wnt inhibited tissues. So we think that potentially Wnt is upstream of this dormancy and stem-like feature. So in summary of this work, what we've been able to show is that KRAS mutant cells and P53 mutant cells are outcompeted. So this substantiates what we'd seen before, but if the cells have both of the mutations, then they're not eliminated. And we've got some data to kind of suggest that they're also not detected and that there's no change in any of the cell biology when double mutant cells are in contact with normal cells. So we think that cell dormancy and cell cycle arrest protects against cell elimination. I didn't have time to show you the data on this. Using our in vitro assays, we were able to show that if cells are arrested in the cell cycle, then they're not going to be eliminated from the epithelium, suggesting that, again, cell cycle arrest protects against cell elimination. Of course, whether this is happening in vivo remains to be determined. We know that Wnt5a is activated in these tissues, and we think that Wnt5a is stabilizing E-cadherin-based cell adhesion and is protective, again, allowing these mutant cells to avoid cell elimination. Our data certainly suggests that stemness, or being stem-like, is a winner phenotype. So that kind of fits with the cell competition model, that being able to retain a stem cell characteristic allows you to be a super competitor, or allows you to avoid being a loser for certain. But our data also suggests that there are Wnt independent mechanisms. And as I said, that's really our next port of call. But I think what I'd really like to try and convince you is that KRAS mutations are not sufficient to drive cancer. And it really depends on which cell that is expressing this mutation and the context and in which this cell is placed. So what is the neighborhood like, whether it has to compete to be eliminated or avoid elimination? And these are all kind of questions that we're hoping to pursue in the future. So just to summarize everything, so what we've been able to show is that when RAS mutant cells are present in tissues in low numbers, they're very often eliminated as losers, and this can be through a process of cell extrusion in simple epithelia. We know that modulation of E-cadherin is really important, and this is driven by EphA2, and it's reversed by Wnt5a. And of course, the next question is how are these two signals connected? And we don't really have an answer to that yet. But certainly it's quite intriguing that the EphA2 knockout phenocopies the Wnt5a phenotype. So potentially there is some direct relationship between these signals. So being able to survive really depends on being able to activate this Wnt5a, being able to stabilize your E-cadherin, but also moving into a cell cycle arrested state or a dormant state. So what controls elimination? We still really want to unpick this, especially in the complex tissue of the pancreas. And this is a project that we've just started where we're looking at tissue location. So where are these cells residing relative to the tissue architecture, relative to the anatomy, so head, body, tail, for example, but also relative to structures like blood vessels, islets, ducts, etc. And whether we can then predict whether cells are going to be eliminated or not, depending on where they're positioned in the tissue. And we're also testing the hypothesis that being a stem cell is a winner phenotype, and potentially that either because you're a stem cell to start with, you're more likely to be left behind in the tissue, or what would be even more intriguing is that having to compete to stay in the tissue programs you to become more stem-like. And what does this really mean for cancer? So how do cells that survive then exit this kind of dormant phenotype to then expand? And this is something that we're also exploring. So Bea has been doing this in the context of chronic pancreatitis. And I've also had a PhD student who's looking at this in the context of high-fat diets. So this is very early days, but what we're hoping is that we could maybe unpick some of the very early events that need to happen to allow cells that are surviving but not really proliferating to then go and expand and go through transformation to pre-malignant stages. So with that, I'd like to thank everybody in my lab, particularly Bea Salvador, who's just coming to the end of her fellowship. And she's going to be a junior PI in the pancreas field, so definitely keep an eye out for her. Non Williams is my new postdoc on the new project, and my alumni, especially Will Hill, Sean Przansky, and Andreas Zarakoulias, who basically established the low-dose tamoxifen cell competition model in the pancreas, and were able to delineate the requirement for EPHA2. And my collaborators, particularly Jen Morton, Alan Sampson, and Maria Sakrir at UCL, and my funders. And thank you very much for listening, and very happy to take questions and feedback. Thank you.